Hello, everybody. It gives me immense pleasure to introduce Dr. Corina Tarnita as the graduate student invited speaker for the Spring EPB seminar. If I started listing all of Dr. Tarnita's accomplishments and accolades, we will be here for a long time, so I'll keep this brief. Dr. Tarnita is a professor at Princeton University in the Department of Ecology and Evolutionary Biology. Before joining the faculty at Princeton, she was at Harvard, where she accomplished her PhD work on evolutionary dynamics in structured populations, where she also was a postdoctoral fellow in the program for evolutionary dynamics, and later was a junior fellow in the mathematical biology. Her research has been funded by many prestigious organizations, including NSF, Templeton Foundation, Alfred Sloan Foundation, Moore Foundation, and others. And her research has also received press coverage by The Atlantic, BBC World, Scientific American, The New York Times, CNN, the list goes on. Dr. Tarnita's research spans many different systems, from microbes to large herbivores in African savanna, as you can see from a very interesting list of publications. With that, please join me in welcoming Dr. Tarnita for her talk today. Over to you, Dr. Tarnita.
All right. Thank you so much for that very kind introduction and for the invitation. It is always such a great honor and pleasure to be a graduate student, an invited speaker of the graduate students. It's such an honor that I never say no to such an invitation, and I'm absolutely thrilled to be virtually visiting. As you can tell, it's the first time I'm actually giving a virtual talk from my office, so I haven't figured out exactly which screen I'm supposed to look into, but hopefully we'll manage and it won't be too distracting. So with that, so one of the questions that I am most excited about and most interested in has to do with biological construction, the way, and especially the kind of construction that takes very similar units, puts them together, and a new kind of organizational unit emerges. What has been called major hierarchical transitions in evolution, such as the evolution of multicellularity or the evolution of eusociality. And when I think about those, there's no, there are endless beautiful images that one can come up to showcase the extraordinary beauty and complexity that social social groupings and collectives are able to achieve. I think I mostly show this image now because I find it so striking that the Sagrada Familia looks so similar to these magnetic termite mounds. I'm not sure actually if they're the cathedral ones. So in any case, I think there's no denying the kind of complexity that that can be achieved once things are social, but certainly, as soon as we think about it, all of these origins share one really important thing, and that's that they all start small. There's no way around it. They all start in small groups, and those small groups face very similar challenges. First and foremost, those small groups have to persist and displace or outcompete an ancestor that has been making, or a solitary ancestor that has been making a living in those places for a really long time. So there's always, the kind of work that I do tries to boil down the question of thinking about these transition to these very origins and try to understand how exactly, what makes some groups succeed so spectacularly, but so rarely, and to have achieved these kind of major transitions, whereas so many others have either stayed in a very primitive kind of form or have altogether failed. There is one other thing. Certainly, these groups have to displace they have to do well enough to outcompete this the ancestor. But they also, as soon as we think about these groups as basically assemblages of very similar units and very small to begin with, because they all start from one or a small group of individuals, then we have to wonder, what makes these groups robust, how do we get any kind of complexity in them if they all start from a small number of individuals and it's a very and they're all very, very similar, possibly genetically identical, depending on what we're thinking about. How can you get any kind of complexity arise, and also how can you get any kind of diversity? How do we get the kind of diverse life cycles that have that we observe today? So in thinking about these questions, when I was when I was getting interested in thinking about questions in biology, the perspective, or the way my inroad into thinking about such questions came very much from with a top-down kind of view. That looked at these groups as, well, the groups have to form and they have to persist. The groups have to be maintained somehow. And certainly one really bad thing that can happen to them is if they have cheaters inside them. So there have to be some mechanisms that maintain those groups, that ensure their robustness. And if they can be maintained long enough, so we have the evolution of some sort of cooperation, and that cooperation can persist, then eventually we might get integration and differentiation and some various forms of complexity. And so this top-down approach that's basically looked at the extent multicellular organisms and eusocial beings, but primarily social insects, have been really informative in trying to distill and compare across major transitions and try to propose evolutionary hypotheses for what might have mattered at the origin. And I spent quite a bit of my early career trying to think about mechanisms. It's typically hard to think about this integration step. That integration step has always seemed to me as a little bit philosophical and magical. It's never really, it never was extremely clear what exactly needed to happen there. But it's certainly easier than to focus on the first part and ask, how do you maintain cooperation? How do you evolve cooperation? How do you control cheating? And so I did quite a bit of work early on trying to understand mechanisms for the evolution of cooperation, and one can come up, and many others have done lots more work than I have in that general area. But then you end up with proposals like you need some sort of there are mechanisms that have to maintain that cooperation, and a very universal one is assortment with like individuals, often kin individuals, so genetic relatives. And then you start to focus on things such as unicellular bottleneck or other ways to control cheating. But over the last 10-15 years, there have been some really exciting discoveries in comparative genomics that have kind of motivated me to start thinking about a complementary sort of alternative approach perspective on these major evolutionary transitions. So recent work in comparative genomics has produced two types of data that have somewhat changed and enriched the perspective on how to think about these major transitions. So one of them is that we've been accumulating evidence for the important role of the ancestor, the complexity of the ancestor, the possibility for co-opting some of the functions of the ancestor, the things that the ancestor was able to do in its own before it ever became a group, might then get co-opted to do something new. And so in the context of the multicellular group or the social group. And so accumulating evidence from comparative genomics precisely focused on the origin of multicellularity, and especially work in choanoflagellates, from Nicole King's lab, also work from Iñaki Ruiz Trillo lab. They've been really, really instrumental in showing that basically roughly 80%, let's say, of all of the functions that we thought were novel inventions of multicellularity had actually already been there in the unicellular ancestor in some form, and they have either been completely taken and translated or co-opted in some way to do something novel in the context of the multicellular organism. And so that's been a way to simply say, Yes, this ancestor has to kind of cooperate ultimately, but maybe instead of just thinking about the ancestor as this, otherwise pretty naive object that has to cooperate, we should think about it as a possibly much more interesting, much more complex creature that then can actually, how complex it is, can translate into spontaneous attributes of these early originating groups that we might have overlooked. And that might change how we pose the questions that I outlined at the beginning. The other really important part of the comparative genomics work, and this was work done, for instance, in slime molds by Pauline Schaap, has shown that it's kind of disproved the widely assumed idea of a certain intuitive order, that things go from simple to complex. And so that's also been, I think it's changed a little bit how we think about these theoretical and conceptual frameworks. So I want to present today 3 vignettes that basically all have in common the idea of looking at these origins of both multicellularity and eusociality from the perspective of just considering the ancestor a little bit more and considering the potential, the newly forming group at the origin in the context of the life cycle of the unicellular ancestor, right? The group emerges as a stage in the life cycle of the unicellular ancestor. And so if we put it into it, can we come up with new insights? So part of the work will be theoretical, part of the work will be experimental. So hopefully there will be a little bit for everyone. So I'll start with the question of complexity, partly because the question of complexity has always been, it's always been this sort of, how long might it take to get complexity to emerge? So one of the earliest papers that I wrote was a collaboration to think about the evolution of eusociality and the modeling question. And so the eusociality is the social behavior in the very special social behavior, sorry about that, in social insects. It's a very special social behavior that requires, in addition to having a social group, there have to be overlapping generations, and there has to be some sort of division of reproduction. But we turn the question into a broader question. And so the modeling question that I want to focus on right now is the following, and can be applied easily also to the origin of multicellularity. So if a mother was able to make some daughters that stay at the nest and help, would that be favored by natural selection? So we want to contrast what we thought of as the solitary ancestor. This would be an ancestor that would, if we think of it as a solitary wasp, would deposit an egg, provision it with some food, maybe stick around for a little while, but then leave and do the same thing elsewhere and elsewhere and elsewhere. So definitely a solitary kind of strategy. And we thought about a very simple eusocial, so something might have happened that now allows this mother to make some daughters that stay at the nest. And it's a very, very simple setup, but basically a fraction q of the daughters stay at the nest and a fraction 1 minus q go away. But the daughters also inherit this mutation or series of mutations that makes it so that when they go away and they start their nest, they also have the ability to retain q of their fraction q of their daughters at the nest, and so on and so forth. And so this allows both the formation of larger groups from the daughters that stay at the nest and the propagation of the behavior from the daughters that go away and start their own colonies. Now, these two different lifestyles would have coexisted. So again, it's the same idea that probably the eusocial mom is not very different from the solitary one. It's probably still eating the same things. It's eaten by the same things. It lives in the same area, so they are competing for space. So the way we modeled this is basically as a competitive exclusion. We didn't conceive of the fact that these two strategies could coexist, but one had to be better than the other and displace it. And the question was, under what conditions, what kind of benefits might exist to offset whatever costs might exist for the eusocial mom for those colonies to persist and displace the solitary ancestor. So it's a very simple competitive exclusion model. And the model prediction, if we were to just think about it in a very simplistic way and think about a summary of the benefits and costs as the per capita growth rate on the y-axis, and it's left precisely as a per capita growth rate rather than the queen reproductive rate or whatnot, because individuals could still all reproduce. We can apply it, as I said, also to multicellularity. It doesn't have to necessarily have anyone be infertile here at the very origin. It's probably fair to assume that the social mother, the eusocial mother, would be paying some cost early on, because she has to not only lay the eggs and provision them with the first bit of food and maybe stick around for just a little while, but she has to just stay there and actually rear them. And mind you, probably in the beginning, it happens sort of one or two at a time. And so this group grows relatively slowly, so that the original mom still has to do quite a bit of work, and everyone does. Eventually, probably this colony will get to be like, if it had enough individuals, it will do really well in terms of the growth rate of the whole colony as a function of the colony size. But at first, it will probably not do so well, because everyone has to do a lot more work, and they're more subject to predation and so on. And now the prediction of the model, there were several aspects of it, and all of the benefits and costs have to align really well. And there's a pretty narrow window where eusociality can evolve. But the more important part of the prediction that actually worried me quite a bit at the time, because I wasn't even sure exactly how to think about it, was that no matter what your benefits and costs was, the most important thing was that those benefits had to arise at very small group sizes. So the benefits of being in a eusocial group had to kick in at small group sizes that at least in the way I was looking at the calculations then, it was 10 individuals, a handful of individuals basically. And so the question was, why would for per capita growth rate in a group of 10 individuals to offset that of one individual in some significant way, that meant that somehow that group had to harness, something had to happen within the group. I didn't really even have the right language to think about it at the time, but I would say in retrospect, somehow the group had to be more than the sum of the parts. But mind you, again, a very small group of a very similar individuals. This is a mother and her daughters. So very similar in size, very similar in age, genetically very similar. So what would they harness in such small groups to be able to act as a mini complex system or some sort of rudimentary complex system? So I didn't really know exactly how to think about it then, and I didn't know, when one works on these origins of things, it's never clear how far one can push the experimental work to try to probe some of these predictions. But in collaboration with a good friend who was, I knew him at the time, and he was a postdoc at Harvard at the time when I was developing with my collaborators this work, and who then started his own lab at Rockefeller University, Daniel Cronauer. After about 10 years of just talking about it, finally, they had this amazing model system in which we could actually test it. And that's like any theoretician's dream. I don't even care if I'm right or wrong. It's just amazing that someone has taken enough interest and has developed a system in which they can probe this. Because whether right or wrong, I will learn something from it. And so the model system was this clonal radar ant, Ooceraea biroi. That's like the way I probably naively think about it is basically like a very primitive army ant. So they are radar ants. They raid others, but they're not sophisticated and huge in their behavior. And the amazing thing about them is that they are fairly primitive, meaning that they in the field, the colonies range from a dozen to 100 workers. They're very easy to maintain in the lab, which is wonderful. And they're particularly easy because they're queenless. And so all individuals, they have a life cycle where all individuals lay eggs at one time. And then as soon as the larvae come out, all individuals start foraging and taking care of them. And so that makes them fairly easy to maintain in the lab, and simultaneously to and to bring back to the lab, but simultaneously it gives you a lot of control, because colonies are entirely made of workers that reproduce asexually and synchronously. So all individuals are genetically almost identical. They emerge in discrete age cohorts, so you can mix them and have them be of the same age. And so and they're all of the same size, more or less. And so that makes it fairly easy to basically create the conditions of checking what happens as you increase group size, but do that with groups that are as homogeneous as we could possibly make them, so that they do not have any kind of, to avoid them possibly harnessing anything that comes from having a diversity of sizes or ages or genotypes. And finally, the other really important thing about the system is that colony behavior is entirely controlled by the larvae, which solicit food and care from the workers and therefore induce them to forage. And so it's very easy to then manipulate the task demand in the colony and standardize it, so that when we increase group size, we don't make it easier on any individual worker, so that in principle, the amount of work that needs to be done per worker stays constant. And so in order to do that, we basically maintain a 1-to-1 ratio of worker to larvae. And so what they did, the setup is in that beautiful photo there. They live on these little petri dishes, and they establish their own little nest where they put their eggs and their larvae. And so they put 1-to-1 larvae and workers, and starting in groups of size 1 all the way to 16. And again, 16 means 16 workers, and they are paired with 16 larvae. And then measure the colony growth. So what we saw when their data came back, or when they did their measurements, was that one ant by itself with one larva mostly just fails. 2 ants do a little bit better, but not that well. Eventually, 4 ants is a little bit better, 6 to 8 ants, and then you get to 12 and 16, and you are at 1. 1 is basically the maximum that they can reach here, because that just means that they've successfully reared their 16 larvae. So with somewhere between 8 and 12 individuals, you are already reaching the maximum capacity of this group to just take care of it, to just complete its chores. But you can't do that with 4 individuals or with 2 individuals. And that was just wonderful. It was one of those moments where it's like, how is that possible, right? It's a very small number of as nearly identical as you could possibly make them. And we do see a result like this. So that was just surprising. But the most amazing thing was that within the experiments, we could actually, instead of just guessing, we could see what they were doing. And so what boosted them like this was rudimentary division of labor. And I call it rudimentary, and we called it rudimentary, because if we had focused on it and tried to assess it from the perspective of the division of labor that you might find in a leafcutter ant colony, right, where you know that they have differentiated castes with morphological differentiation, and everyone knows what they're doing and so on, this would not have looked impressive. When you have just 10 individuals, they're not going to be able to this one is going to be a forager, this one's going to be a nurse, this one is going to take the trash out. There's just too much to do. Everyone has to do a little bit of everything. So they're going to live on a continuum. So then, but something does happen at around 8 individuals that starts to look like a rudimentary division of labor. And what becomes conserved is not the specific role of each ant, but the relative ranking within the colonies. So if I am more forager-like today than Israt, when we are 10 individuals, tomorrow I might do a little bit less foraging, and I might have to do a little bit more nursing because there's more demand for that. And she might actually end up doing a little bit more foraging. There's a lot of fluctuation in these very small groups, but tomorrow I will still look more forager-like than she does. So our relative ranking within the colony, not the amount that we do, but the relative ranking, is going to stay conserved. And that starts to happen again at around 8 individuals, and that gets carried over life cycles. So even when the next life cycle happens, these roles will get maintained. And that was very exciting, because this is one of the things about working on the origins of multicellularity, the origins of eusociality. It's very hard if you just think about the metrics that we're used to from observing these like highly successful, highly evolved colonies or multicellular organisms. It's hard to really imagine how it would have looked in these very, very incipient groups. And so this was just a very beautiful example of that. But the other thing that, and I think I might actually, sorry, no, actually, I will tell it here. Sorry about that. One of my graduate students led the parallel modeling that happened in this system. And one of the questions was, how does this rudimentary division of labor actually happen? And there was a very simple model, a model that's been proposed for about 20 years for how specialization happens in ants. We basically retailored it and applied it here, and we could very easily get the level of specialization that we see in these experiments with that simple model. And the most important takeaway from that model was that it did not require ants to actually communicate with each other. So we were able to get the same level of rudimentary division of labor without by relying simply on internal thresholds that ants use to respond to environmental stimuli, such as begging from the larvae or trash piling or whatever stimuli might exist. But they did not need, the model did not require additional social communication between them, meaning that the way I think about it is kind of like when I was in college, I used to live with this group of people who all had completely different schedules. I mean, some of us were night owls and others were really morning people, and nobody really kind of saw each other that often, but we all managed to coexist. And, dishes might be piling up and they would have to be dealt with, and one individual would have a slightly lower threshold for seeing the sink getting more and more disgusting, and then that individual will start doing that work, and then others who weren't even upset by the disgusting looking sink are definitely not going to be bothered anymore when the level of how many dishes have piled up is going down, because someone has already been started to do it. But those individuals might be more sensitive to the fact that there's no more orange juice in the fridge or whatever, and they might go and take care of that. And we didn't have to actually establish who was going to do dishes and who was going to do what. We didn't even have to see each other to basically manage to more or less successfully coexist in that environment. And I think the reason I went into this detour to kind of explain this intuition is because, as I've been discussing with colleagues who work on the evolution of multicellularity, for example, I was talking recently to Iñaki Ruiz-Tillo. He was saying that whereas so many of the things that we find in multicellularity one can find their counterpart somehow in the arsenal of the unicellular ancestor, communication doesn't seem to be one of them. Communication seems to be something that has evolved sort of after the group has formed and is there already. And so this was something that showed that you could get spontaneous division of labor emerging at the origin of the group. So that's basically some sort of rudimentary complexity without the need for actual communication. Sure, if we add communication in there, things get even better, we get even more differentiation, but we don't need it to reproduce these results. And so it's been exciting to see that we've been going back and forth between thinking about these major transitions, and now it's kind of going in tandem as we think about the things that would have been there early on and what is the potential then in these early groups, but what wouldn't have been early on and therefore might have taken some time to evolve. Now, how does this rudimentary division of labor actually boost fitness? Why do we get the growth in the increase in fitness? Well, this is where Chris Tokita was the graduate student who did this theoretical work, and what Chris found was that in the model he made the prediction that basically as you increase group size, task neglect goes down. And by task neglect, we mean the proportion of time that any task gets unattended for. And in the model, it goes way down. It goes all the way to 0 already at 16 individuals. But we then measured also empirically, and we found the same declining trend, although perhaps not as dramatic as, or definitely not as dramatic as in the model. But the point is that basically, the more ants you have, the less likely it is to find any task that is being unattended. And one of the things that does is that it increases homeostasis in the group. And so one of the immediate indicators that we've, in terms of fitness that we found, indicator of this homeostasis was the fact that larvae in groups of 16 individuals developed much faster, like several days faster than larvae in groups of size 6 individuals or 4 individuals. So the main takeaway, I think, we've been doing a subsequent work, and I'm happy to talk more about it, is the other thing that's now really cool about this system is that we were able to also make groups of individuals that are, like, start to make controlled heterogeneous groups and try to introduce different forms of heterogeneity, genetic heterogeneity, and heterogeneity in size and in age, and start to see how all of, like, the interplay of all of them, but in this very controlled way, and then assess what it does to this division of labor. But for the purposes of this talk, I just wanted to conclude this first part by saying that the main takeaway for me has been that we can get the evolution of these social behaviors, but it's very hard because you have to really, these very small groups of very similar individuals have to get a very significant boost at small sizes. And not everything seems to be able to do that. There needs to be something that's there in the ancestor. There has to be some sort of latent ancestral potential that may appear in very different guises for very different species and taxa, but that facilitates some sort of self-organization that can happen spontaneously without needing, you know, to evolve and do more complicated things over longer periods of time, but that can be there almost spontaneously as soon as you have 10 individuals or something of that magnitude. Okay, then I'll briefly talk about the more, this part doesn't have any, I'll just preface by saying that it doesn't have any experiments. So this one is basically continuing on this idea of thinking from the ancestral perspective, and thinking about how diversity actually arises. So how long do we have to wait to get the diversity of life cycles that we see? How complicated is it to get various types of life cycles? And here we pretty much started from the ancestor and thinking, you know, let's take on board all of this work that comparative genomics has been doing. We are focusing on the evolution of multicellularity here. So instead of just starting with a boring ancestor that can't do anything, but we expect it to figure out how to cooperate, let's try to give this ancestor the ability to do something. So here we give it a gene regulatory network that allows it to sense its environment and respond to it. And the problem that this particular individual has to solve is that it lives in an environment that alternates in a very predictable manner. It's environment 1 for the same length of time as environment 2, and then environment 1 and environment 2. So it's really as boring as it can be, other than just being one environment. But it needs to express gene A in environment 1 and gene B in environment 2. And if you just allow this ancestor to kind of learn how to live in this environment, it will eventually come up with a stochastic switch that allows it to just flip back and forth between these two different things. And it is in this context of this ancestor that what we now do is we add group formation. And the way to add group formation is to think about two different kinds of mechanisms by which group formation can occur. One is that when cells divide, they don't fully separate, and they form, you can think of it as forming a simple filament. And this is very much the way of modeling. This is very much the analog of the ants, because instead of thinking them as little groups of ants, we can think of those as simple growing filaments. The other way of doing it is via this coming together or aggregative development, where you have individuals that stick to each other. So now we have an ancestor that is able to live in its environment and do something there, solve a problem. And then we have two ways of possibly constructing groups. And the way we did it in this particular model is we said that all of a sudden something might change about this ancestral cell, so that when it expresses gene A, that promiscuously gives rise to cell addition. In other words, it just becomes sticky. All of a sudden, maybe something has changed in the environment or something has changed in the cell itself, but when it expresses gene A, it also becomes sticky. It is not something that has been selected for, it's just piggybacked in some way, or it has been triggered by something. If that's the case, now we have sticky cells. And sticky cells can form groups. And so the very simplest start of this model was to assume that we have a staying together dynamics. So this clonal development, not the aggregative development. Cells will reproduce. There will be a mother and a daughter that are very temporarily stuck to each other. If the cells themselves are not sticky, for example, because they're expressing gene B and not A, then the two cells will then immediately fall apart and division will end. But if the cells are sticky, depending on how sticky they are, they might actually be stuck to each other and start to grow a group. And eventually you can think about how cells might be released. So when cells lose stickiness, then those cells might be released from the group, and so on. So you can think about both the formation of the group and the fragmentation of the group when cells lose stickiness. And we put a very simple, we've been doing some really, I'm super excited about this work because it's happening right now. At the time, we thought this was complicated enough. And so we put a very simple cost and benefit thinking on it. So the benefit, we thought of it in terms of predation. If you get big enough, you won't get eaten. Anything that's below a certain size will get eaten. Anything that has just surpassed that size will not get eaten. We think of this in terms of phagocytosis, right? It's not, the predator is not something that can take bites and eat you slowly, but rather something that needs to engulf you. So if you've gotten big enough, that's great. And that's the benefit. The cost, we thought of it in a very simple, it seemed extremely artificial at the time, but actually we're kind of reproducing this now in a more mechanistic way, and that's the part that I'm very excited about. But basically the cost that we thought about was that these cells will have to compete for space, and they'll have to compete for resources. And so that cell division rate might decrease with group size. So per capita cell division rate might decrease with group size. So it's very costly to be, and we put a very costly function there. It's a very steep cost. But we wanted to see the evolution of multicellularity, and if we can get it in the face of a very steep cost, then we're in good shape. Okay, so now we've introduced in this model basically the groups will form promiscuously because of that stickiness, but now there's the possibility for group formation. And now there's a potential tension between selection on ancestral function and selection on promiscuous stickiness function, right? You might want to escape predation all the time, but that will mean that in environment 2, if you're not expressing gene B, but you're continuing to express gene A so that you can stay as a group, then you lose the ability to, then you actually don't get the full benefit. Your reproduction will suffer because you won't be expressing the correct gene. So the reproductive rate of the cell depends on the ability to correctly express gene A in environment 1 and gene B in environment 2. So we've introduced this tension between what the ancestor needed to solve and what the group is now able to solve. And what we found with this very simple model, we had basically two parameters that we varied. One was the maximum group size, this K, at which you get the minimum group size at which you get the predation benefits, and the lack of predation benefits. And the other was the stickiness parameter, just how sticky you become, because how sticky you become gives us something about the size. It gives us influences how big these groups will grow and how long-lived they are. And so we have K on the y-axis, we have S on the x-axis, and what I'm showing here is that in different areas of this parameter space, we're finding different life cycles that evolved. And already just in this very simple setup, we find 4 different kinds of life cycles. One is a very simple one is the green one. The green one really doesn't want to be a group. It is not getting enough benefit. It cannot grow big enough because it's not sticky enough. It can't grow big enough to achieve a group of large enough size to get the benefits of predictions because it's a very high K. So what you notice is this in environment 2, it has no problem. It just expresses gene B. It's not sticky. It doesn't form a group. But in environment A, it's this tug of war between wanting to express gene A so that you can get the reproductive benefit of expressing the correct gene in the correct environment, but at the same time, not really wanting to grow, because then you start to pay a cost for being in a group. You pay the cost of reproduction without really ever getting big enough to get to the benefits of escaping predation. And so what you observe is one that keeps turning on and off gene A during environment 1. That's a pretty boring one, but I just wanted to start there to give you a sense of how to read these. The other one would, for instance, be the pink one or red one. That one is very much like the metazoan life cycle. That one expresses gene A always. And in fact, the only way to ever get single cells there is when every once in a while, unless you are at stickiness level 1, which means you never break away from the group, every once in a while, probabilistically, one of the cells will simply be peel away from the group, simply because there will always be such a probability unless your stickiness is exactly equal to 1. And so that one grows very, very, very large groups. And every once in a while, it produces a single cell, and that single cell immediately starts to grow again because it is also because it is also sticky, because it is always expressing gene A. So this is just to show that basically, with a very simple setup of an ancestor that's able to sense an environment that's not very exciting, but it's at least a fluctuating environment, and with very simple cost and benefit functions, you can get 4 different life cycles already basically at the origin of multicellularity. And they're not nonsensical cycles. So for example, this metazoan looking like life cycle that I just described. There's also this 4th one that basically is express gene A all the time, except for one point where you don't, just at the switch between environment 2 and environment 1, where all of the cells collapse, but then immediately they express gene A again, and then immediately they start building groups. And that is a little bit like thinking about the slime molds, right? You just form a group, you form your group, you form your group, and at the end of that, everyone collapses, becomes solitary again, but then eventually they become groups again. Now, I'm going to skip a little bit quickly over this in the interest of time, but if in addition to this staying together mechanism, so right now the only way to form groups is when a cell divides, the daughter will stick to the mother and so on and so forth. If you also add coming together in a very simple way where you say single cells might bump into groups and get stuck to them if they're sticky. So we're not merging groups, we're not doing fusion of groups here. Simply single cells might have some very small rate at which they might bump into groups, and if they do and the cell is sticky and the group is sticky, then you're going to stick to the group. If we also add that and nothing else changes, they're still staying together, but now there is this additional coming together, then what we find is 2 more life cycles that emerge. And those 2 life cycles, I will draw your attention to the yellow one because it's particularly interesting to me. That one is one where you express gene A all the time, so you're always sticky, except for the shifts of the environments. And so when 2 changes into 1, you turn it off for one bit, and then you turn it back on again for the entirety of environment 1, and then you turn it up one more time, or turn it back to gene B, and at the switch between 1 and 2. Okay, that means that these groups grow for almost the entire length of an environmental of one environment, then they collapse, and then they rebuild again and grow for the remainder of that environment, and then they collapse again, and then so on. And so in some sense, if we look at these life cycles and we compare, this life cycle 6 is actually shorter than life cycle 5 or 4. So in some sense, these cells build groups, and then they fall apart, build, fall apart, build, fall apart, fast on a faster time rate than the ones in 4 and 5, for instance. And the reason that that was interesting is because coming together has always been, or I've learned about it, as always associated with this seemingly transient multicellularity. You form a group, but then you kind of fall apart. It's never led to forming, human beings so integrated multicellular stage that then remains like that and doesn't collapse to single cells anymore. And I think much of the reasoning for that has been that it's because coming together, it can be costly. It can allow cheaters in there, and therefore it would be very hard to get it to be as robust at maintaining cooperation as you might be able to do with staying together, where it's always one genotype and then it might go through a unicellular bottleneck. And so this is showing that without any costs, there are no cheaters in this model. Without any cheaters, when you introduce coming together, you still get shorter life cycles. And the reason for that is that coming together in this case is not a cost, it's a benefit. In fact, life cycle 6 is a lot better than life cycles 5 and 4 because it can afford to turn gene A off twice and therefore harness the benefits of being correct about expressing gene B twice in that same time interval. The reason it can do that is because coming together helps a group grow faster than just staying together. So instead of just dividing very slowly and growing your group, now you have a bigger chance of doing it because you're also like attracting or bumping into other single cells that can make you grow bigger. And so in some sense here, we observe the evolution of shorter life cycles as a kind of positive, coming together facilitates the evolution of shorter life cycles rather than the opposite way of thinking about it, which would be that shorter life cycles are a consequence of the fact that coming together can't maintain cooperation for long enough. So in that sense, this like bottom-up approach has been really good of trying to come at basically producing alternative ways to thinking about some such statements that from a top-down perspective seem very reasonable. We know that most complex and advanced forms of multicellularity have always come about via staying together. So it must be, and that coming together is always associated with some sort of transient grouping. So maybe the reason is that you can't do better with coming together. This is saying that in fact coming together might just facilitate you to do something that you weren't able to do with staying together, and which is to be shorter lived here. Anyway, so with this, the conclusion of this part is that diverse life cycles and life histories can easily emerge at the origin of multicellularity, basically shaped by ancestral constraints and ecological conditions. And simply that, doing this bottom up can give really interesting ways of producing alternative hypotheses and testing existing hypotheses. And finally, I'll end with what makes construction robust. So when I started at Princeton, I was very, one of the things that had been on my mind was that if I could do experiments in any kind of system, it would be in a system where the groups, that there isn't some sort of perfect kin recognition there is aggregative multicellularity, again, because this aggregative multicellularity always seemed kind of like an imperfect way of doing it for a variety of reasons. And where aggregative multicellularity, the organism that does it, isn't able to fully recognize and reject non-kin. And I had toyed with the possibility that maybe Bacillus subtilis would be a good example, and I didn't really know, and no one at Princeton was working on anyway the kind of setups that I had in mind. And I happened to be the question there, but what if you're unable to do either of these, meaning what if you can't recognize kin or eliminate the possibility of non-kin from joining the group? And then this model organism has just like fell into my lap in the most unexpected way. And I have to thank Joan Strassman for that, because I was at a workshop, I think at KITP, and she gave a talk and she started to tell the story of this whole, I mean, I had known about Dictyostelium discoideum, but what I didn't know is that it was doing this. And this is, there's a large diversity of coexisting strains in nature. They, on very small spatial scales, which means that they probably eat together and starve together. And it seems that chimeras form readily. Mind you, there is some kin recognition that exists, but it's not perfect. And so there's been some work on that, but chimeras can easily form with strains that have been picked up. And I think Joan and Dave's group had by that time already accumulated evidence that if you simply just pick up fruiting bodies from the wild, you are going to find chimeras. So chimeras actually form not just in the lab, but also in the wild. Maybe not every pair of strains can do it, but certainly there will be some pairs that can. And when that happens, and so they were interested in, I think, several groups here, but I think Joan and Dave were involved in many of these, maybe all of them. When you bring them back to the lab and you set up a very natural experiment to see whether you can find reproductive skew or not, you basically start with 50-50 cells of two different strains, and you let them aggregate. They are starving. You let them aggregate, and then you look at the spores, and then you see whether you find 50-50 of each strain. And if you don't find 50-50, that means that one of them was better able to put themselves into spores, and so you might be able to detect whether there is any kind of skew and the possibility for cheating. The interesting thing was that there was definitely reproductive skew in basically every pairing, and that suggested that there should be the possibility for rampant cheating. And immediately the question rises, how does sociality persist despite this apparent cheating? The even more mind-boggling result was that at least these early studies showed that there was a linear dominance hierarchy, so that genotype A was always better at getting into the spores than B, and B than C, and so on. And so that also immediately made the question of how does sociality persist equivalent to a similarly important ecological question of why on earth do we still find all of these strains in nature? Why do they coexist? Why isn't one, the dominant one, taken over everything? And there's that. And so I think also at that same time, and I think pretty much in the same talk, I made the following I made the following observation that then I spent basically years pursuing, because we were not equipped to do these experiments. I don't have a wet lab, and so we've done it in collaboration with Thomas Gregor's group here. He's in the physics department. He's a biophysicist who already had Dicty in the lab. So here's a video of the aggregation of this, and I'm sorry, I got carried away, but I should have said that basically what Dictyostelium discoideum does is that they are free-living amoebae that they feed on bacteria, they live in leaf litter and in the soil, and when they starve, they undergo this major coordinated process of aggregation that eventually produces a mound, and that mound ultimately becomes a multicellular organism that has an inside-outside boundary, and it can crawl on the plate and so on, and can do chemotaxis and phototaxis better than any other single cell. And ultimately, when it finds the right conditions, it hurls itself up and it creates this fruiting body where about 20% of the cells die to produce a stalk, and about 80% of the cells become the spores that then go and are dispersed or are anyway resistant to starvation. And once they get to the right kind of environment, then they switch back to being solitary cells, and they start to eat and divide and carry on as solitary individuals until they starve again. So here's a video of that, but focused in such a way that you actually see single cells. And the thing that I became obsessed with, here you see the aggregation. It's wonderful, it's gorgeous. You won't see much of it because the focus isn't that. The focus is these individuals that simply do not seem to react to what everyone else is doing. They're just doing their thing. And we call these loners, I guess they're simply individuals that do not seem to be aggregating when everyone else is. And the reason I got really excited about this is that I started to think that what if we think about the full life cycle of this, the full life cycle is not what I showed you on the previous slide, but rather it also involves this separate life history strategy that stays as solitary individuals even though they are starving. And the reason for that, I thought, from an ecological point of view, is that it could be like a bet hedging strategy where these ones stick around just in case food comes back immediately in the environment. They're actually pretty long-lived. They live for about 200 hours, and so they can persist. Eventually, they'll die, but 200 hours it's actually a pretty long time, and especially if you think about their nutrients being, whether a deer passes by and drops some dung, and then they have a feast for some long period of time. That is pretty uncertain, but once that happens, these individuals could take advantage of the food immediately, whereas the others are completely committed to the multicellularity process, which is irreversible, and so they can't stop, break away, and start eating. So these ones get quite a bit of a benefit from being able to eat immediately. So I thought a bet hedging strategy could make sense. And if you live in like areas of the forest where deer don't go very often, then you might be more inclined to produce a lot of the spores, because it would be a bad idea to leave too many of these individuals behind. But if you live in areas of the forest where deer hang out a lot, you might actually not make that many spores. You might make some, but you might also leave behind a lot of these. And because these disperse all over the place, we might find a mixing of them all over. I will make the long story short here, that after this is that, of course, the first step was to figure out whether these were, first of all, viable. Are these cells actually able to do anything? And the answer is yes. If you give them food, after you've picked up the fruiting bodies off the plate, if you give them food, they eat, they divide, and their progeny are perfectly able to recapitulate the multicellular process. So that's great. But the more important and much, much harder to answer question was whether they are simply random byproducts, right? If we talk about synchronizing and coordinating millions of cells, sometimes, right, you can't make an omelet without breaking some eggs. You can't like make an enormous, a million cell multicellular organism without leaving some stragglers behind. So if they were just random, they would be quite boring, because then they wouldn't be a life history strategy. None of that would be happening. But we hoped that wasn't going to be true. And finally, my extremely talented graduate student, Fernando Rossine, was able to set up just the right experiments to test whether they were whether they were random byproducts. And these are experiments that show what happens as you increase how many starving cells you start with. So that's the initial plated cell density. And I'm going to look at the loner density. And my naive prediction was that, well, first of all, the correct prediction is that if you don't have enough of these, they're not going to be able to do their quorum sensing. So at very low densities, everyone is going to stay as a loner. And that is a correct. But eventually, if you get a high enough initially plated cell density, the prediction was that maybe the way that these loners form, ideally, would be, if they are indeed not random, but part of the life history strategy, maybe it's a stochastic switch, so that they flip a coin to decide who stays and who goes to become a spore. So I made this prediction. And what eventually Fernando found was that they're for sure, but this part was correct, they're for sure not random, but they're also not a stochastic switch. They actually turn out to be a much more interesting strategy. These are loners that the decision to become a loner is not a context independent process, it's a context dependent process. And we eventually demonstrated that different strains, so first of all, it's a conserved quantity. So it's the number of loners that stays constant, not the fraction of loners. Different strains, here red and pink, red and blue, have different numbers of such loners that they leave behind. But notice gray is not different from red significantly, though it has much higher variance, and there's a whole interesting story around that too. And we discovered that they are very sensitive to a lot of things, such as agar concentration. You have more porous agar, you get fewer loners. You have less porous agar, you get many more loners, and you increase the differences between strains. So they're also sensitive to interactions with other strains. So the loners became this whole thing that instead of being sort of, first of all, they're not broken, they're not random byproducts, they seem to be part of the life history strategy, and they seem to be doing that in a very interesting way that is a context-dependent process. The loners, in some sense, are almost kind of like we are all loners right now, by like a social choice, not by a individual choice necessarily, right? So the loners are part of the whole process. And so we made a simple model that I will actually skip through. But the loners, the takeaway is that the loners are influenced by both abiotic and biotic factors. There's a very simple model that we made to explain it, and we can actually reproduce many, many features of the behavior, but I'll skip, and if anyone is interested, I'll happily go through it. We can actually prove that the model plateaus, and we have all of these parameters that we've been enjoying playing with, and some of them have experimental tests to prove that they influence in the way that we predict that they influence. But I want to end this part by basically saying that beyond this being a bet hedging strategy, the whole reason that I think it has to do with robustness is because these loners, in some sense, they are not subject to whatever cheaters might be in the multicellular organism. They are outside of the multicellular organism, and therefore outside of the reach of those cheaters. But they can recapitulate the multicellular process. So here's by basically, again, looking broader at the life cycle of these multicellular organisms and finding these other life stages or life history strategies. Here's an alternative to how one can ensure the robustness of a major transition without necessarily saving every group, right? And so the idea would be that some lack of synchronization, these might increase the robustness because these loners are like reserves, like they're like a seed bank. They can always recapitulate the innovation of multicellularity without themselves being subject to the possible cheating and other things that might happen within the groups. And of course, since I've started to do this, then almost in every place where I've talked to people who work on coordinated, large-scale coordination, whether it's the wildebeest or other systems such as locusts and so on, everyone says, oh, we have loners in this system too. And so now we've started to kind of put together all these examples that we've been learning about and try to understand, maybe Dictyostelium discoideum was just the first system where we could actually manipulate things and learn something about the fact that these loners are not just something that's an undesirable byproduct, but might be actually part of the system's, the life history strategy of a system. And so we've been doing some work actually with the wildebeest. That's been extremely fun, and I can tell you about that. All right, so I'll conclude here by basically making again the case for the bottom-up approach as a complement that allows us to add some mechanistic insight to elucidate some specific evolutionary trajectories as they unfold from the unicellular ancestor, and then help to critically evaluate some general evolutionary explanations that have been proposed with the top-down approach. And with that, I thank my collaborators. My graduate students are in orange here, and they've been instrumental in doing all of this wonderful work. And Ricardo Martinez Garcia is actually a postdoc, or was a postdoc, and I would love to take questions if there's any time left.
Hello, yeah, Corina, thanks for the great talk with so many stories. I think there are some questions in the YouTube. Yeah, and yeah, And Israel, would you like to read the first one from Jonathan Losos?
Yeah, sure. Yeah. Thanks for a very interesting talk, Corina, and I definitely will be thinking a lot more about loners now. But first, so Jonathan has a question. He says, great talk, Corina. Your emphasis on the state of the ancestor reminds me of the old debate of the extent to which evolution is deterministic or contingent. Your findings would seem to come down on the side of Stephen Jay Gould, that evolution is not inherently predictable and to a large extent depend on what happened in the past. Would you agree with this interpretation?
I mean, yeah, absolutely. I think that there is a contingency, I think, is a really important element of evolutionary. I'm not sure. I have to admit, embarrassingly, that I'm still, learning about biology. In the last 10 years, I've been mostly just reading. I don't think I've read Stephen Jay Gould enough to absolutely, compare with all of his work. But I would say, yes, I think what we are learning is that there is some potential that exists in the ancestor that I think of it as general constraint, but I don't think of constraint as a negative thing. It is simply just something that constrains a direction that you might take. It takes you in a certain direction more than another. But yeah, I think all of these genomic comparative genomics studies also point in that direction, and I don't think that's fully the story. I don't think that once you have all of those constraints, there is just one thing that can happen to you. I don't think it's fully deterministic, but those constraints do matter. And they matter in this way that they actually give you a lot more to work with at the beginning than we would have realized if we think about it only from the top, right? Like there's a lot of potential that can happen spontaneously, and that potential can be for will take you in some direction or another direction. Yeah.
Yeah, thank you. And actually, before Jonathan Losos, like David Quiller and John Stressman send their greetings. And yeah, and then like we have a question from Callan. He says, thanks, Corina. In response to the evolution of diverse life cycles, to what extent will we see transitions between different aggregation strategies? Are certain transitions more common, likely than the others?
What kind of transitions?
The transition between different kind of aggregation strategies.
You mean in this particular, like in this setup, in this part of the model?
Yeah,
that's my interpretation. Isar?
Yeah, I think he means how you have different kinds of transitions, and some of them are more aggregate-like, whereas some are not. So is there, what extent do we see a transition between the different kind of?
Yeah, I should say that all of these evolve in their own little ecological space, right? So if I have like a really big predator that requires groups to grow a very large size, right? So this very high K here before they get the benefit of not being eaten by predators, then I am more or less constrained to evolving this one life cycle, right, in this particular setup. But if I am in an area where my predators are actually not that big, and I'm actually quite sticky, then I can get a bunch of different life cycles. So I wouldn't think about these as kind of evolving one from the other, but rather from the same starting point, the same ancestor, depending on what kind of predator I might have in that environment, what strategy might I evolve. And the reason that I'm excited about this right now is that I have a master's student who is very keen on introducing geometry here. This is another thing, like when you start to think about how groups are formed from the ancestor and you start building them up, you very, very quickly start to realize that they're not just a sack of cells, right? They're depending on what the ancestral cell looked like and the ecological conditions, we might get very different kind of geometries, for example. You'll spontaneously get geometry. They don't exist in some sort of vacuum, right? and so we're kind of slowly building in these different features, and as we build geometry in there, then we find different restrictions on what life cycles, how these life cycles will form here. So so stay tuned for that. But
yeah, that's exciting. The next question is actually from me, which is that in your example with the ants that you show that you can evolve rudimentary complexity and emergent behavior pretty easily. So, and my question is about the reversal. So in evolution of yeast, they have seen that like in the rudimentary complexity can very easily lead to reversal to unicellularity. So do you think, or for different kind of systems, like when a system is at that point where it has evolved a new emergent behavior, what are the ecological or other selective pressures that might lead to the system going one way or the other, such that they are not, because there are some can evolve back to a simpler stage or can evolve a higher complexity. So what do you think?
Yeah, I think it's a really important question, right? What is the point of no return to any of these? Is such a hard question that has so many nuances, right? I think if I think about these ants, for example, once you have this spontaneous self-organization that doesn't require anything, in fact, it comes for free from whatever this ancestor was able, like this ancestor basically had the ability to sense the environment, and whether it's aware that other ants are also there and doing things in that environment, maybe, but it's not necessarily needing to plan with them, but spontaneously they can now take care of this group, and if they're big enough, they're all of a sudden really doing well, then I think that this group will, in principle, will persist unless something changes. I mean, when we created this very simple model here that didn't have any of the spontaneous organizations, here just phenomenologically, we compared these two different strategies. It was a very narrow window in which we could get eusociality to evolve. Even if you have the spontaneous benefits arising, these benefits arising at very small group sizes, you still need, like you need an ideal window of, in terms of what the queue should be. If too many of your daughters stay at the nest, you grow too big, but you very rarely seed new colonies. So you're inefficiently using the landscape or the resources. If queue is too small, then that means every once in a while you'll keep a daughter, but you'll never get to be the size that you need to be to get those benefits, right? So it was a very small window where eusociality could evolve, and a very large window where the solitary ancestor couldn't. But once you had it, even in this phenomenological model, it was very hard for it to be invaded unless something happens in the system that makes it so that the benefit, so something might change about the queue. Now all of a sudden I do have the sweet spot in terms of the queue. Enough of my daughters stay, but not too many of them, and I'm growing big enough. But something might happen, and all of a sudden, maybe more of my daughters start to disperse, and not enough of them stay at the colony. For whatever reason, then you can lose out again to anything that can be solitary. So, in principle, at this origin, it could be fairly easy to go back and forth, but something has to change, because if you do manage to get like some sort of something happening inside that organizes you and you get these benefits, it would be very hard for any solitary ancestor to invade. Do you see what I mean? As you get more types of differentiated individuals, so maybe you evolve communication in there, and now you get even more specialized, right? At some point you start to it's not even clear what could invade anymore. I was talking to Iñaki, and his way of talking about it, if I can borrow his metaphor, is kind of like asking what will replace my iPhone, will I be back to a flip phone? It's not just a flip phone, right? It's that I have to consider that this has become my camera, my maps, my photo storage, my everything is so much of it is in there. It's not clear what kind of what is the solitary ancestor that can invade anymore. It certainly won't be the first thing, a cord phone or whatever it's called, right? So it becomes a challenging problem to pose, a challenging question to pose even correctly, depending on the stage at which your group gets. So anyway, it's a hard one, but I hope I had some.
Yeah, thanks for the explanation. When you want to ask your question yourself?
Yeah. So, Karina, I have a question. Like, I'm pretty interested in your end research, and you have a term latent ancestral potential. I'm curious, like, do you think that ancestral potential could be the genetic relatedness? I have been learning, like, the genetic relatedness is necessary for the socialism to develop. Do you think that's also necessary for multicellular cellularities? If without the genetic relatedness, could the multicellularity go into parasitisms or the bad side?
Well, I should be clear here. No, I don't think of relatedness as a latent ancestral potential, only because, but your questions, you had two different aspects of the question. That's not what I meant here, because all of these groups they're like this model, basically all individuals are genetically identical. And I'm saying within this model in which groups are formed by staying together with genetically identical individuals, some groups will make it and some will not. And the ones that make it need to have something that allows them to harness the benefits of being in a group at very small group sizes. Okay, they're already all genetic, they're clonal. Okay, the way I'm thinking about them. And then the ants in the experiment are also clonal, right? But only some. So there has to be something about the solitary wasps that allowed them to, if some daughter stays with the mom because maybe she can't fly away, the wings are imperfect, something has happened, right? And they're there, then she'll start helping the mom somehow. And what we basically showed with these experiments is that she doesn't need to want to help the mom. She doesn't need to know that she's helping the mom. She doesn't need to plan it with the mom. All she needs to do is react to the fact that there are other eggs in the nest and start to do the chores that are needed to take care of those eggs, you know. So maybe she sees that there are two larvae that are begging, and so she starts to go for food because maybe the original mother is like now cleaning them of parasites or something like that, right? So all you need is this kind of back and forth, these like different minute differences in their thresholds in how they respond to stimuli, but the ability to basically react to those stimuli. So there must be something in the solitary wasps that allows this kind of organization to happen, but not in the many species of beetles that exist and haven't become eusocial. Even all else equal in terms of, you know, are they completely related, very like clonal, highly related, or whatever else you want to throw in there, right? So all of that already given, what else do you need to push you over the edge, right? Do you see what I mean? So that's what I mean by the ancestral potential. I don't mean whether they are able to form clonal groups. I'm already providing that in the model. And you still don't get it automatically. Something else also still needs to push you over the hump of these benefits and costs.
Yeah, so yeah, I get it. So it's like their kind of native behavior to help each other when they have chance to.
What's that?
It's like their native behavior to help each other when they have chance to. Like you mean the solitary wasp, if they cannot get away from their mother, and they're gonna help her mother or maybe her siblings.
But without necessarily wanting to, right? So this model did not require the daughter to know that it is helping a mother. It simply required every ant in this colony to sense that there's stuff that needs to be done around the colony, right? So it doesn't have to have this like, am I going to be altruistic now? It's simply, if you throw these ancestors into a room to get into like a little nest together, will they react to what the others have already done to modify this environment? Like if one of them has started laying eggs, will the other start to like clean them? But not necessarily because it is altruistically driven or because it knows that it is taking care of some other individual's eggs. It could be that it's thinking that it's taking care of her own eggs. Do you see what I mean? I'm not making that difference here because they're all genetically identical, but both in the model and in these experiments. But that's how I would think about it.
Yeah. Okay. Yeah. Thank you. So I think if there are,
yeah, I think that's all the questions that we had, and that ends us at the right time.
Yeah, that's perfect.
Yeah, I think the streaming has stopped, has it?
yeah, the streaming has been stopped. Yeah.
Okay, cool. Yeah, great.
That's just the delay.
Okay.